Assalamualaikum warahmatullahi wabarakatuh. Hi everyone. Here we want to presentation or discussion about enzym and coenzym. And we are from first group and members of the group there are Anissa Oktasafitri, Keisha Aulia, Naila Desviana, Nurlilis Fitria, Renggi Maulana, and Rini Puspitasari. Next, what is definition of enzyme and coenzyme? First, definition of enzyme. Enzymes are biocatalysts that speed up the rate of chemical reactions in the body by lowering the activation energy of the reaction. Enzymes are chemical compounds in the form of proteins that act as biocatalysts, where bio is living things and catalysts are substances that can speed up reaction. Simply put, enzymes are catalysts that help speed up biological reaction. And definitions of coenzyme. Coenzyme is a cofactor, a small organic molecule that is actually part of the enzyme itself that is resistant in heat, containing phosphate and also ribose. It can soluble in water and can blend with apoenzyme to form holoenzyme. Next, we have function of enzymes and coenzymes. The first is catalyst. Coenzymes have an active function as a catalyst that can increase the catalytic ability of an enzyme. Second, electron transport. In addition, coenzymes also function to determine properties of a reaction and can act as an electron transport from one enzyme to another. The third consists of organic. Coenzymes consist of organic matter and are identical to vitamins because many coenzymes are found in the derived forms of B vitamin such as niacin, thiamine, riboflavin. And the fourth, cell actors. Coenzymes also from other molecules in cells that be a source of cell actors. An example, one of the functions of coenzymes for the body is memory retention. Without coenzymes, the human body doesn't work and also process stop.
Enzyme structure. Enzymes are single proteins or a combination of protein and non-protein compounds that can only be produced by living things. 1. Apoenzyme. A. Active site. This is the site that is attached to the substrate. Substrate is a substance that will be made into a product. B. Allosteric site is the site that binds to the cofactor or activator of the enzyme. The allosteric site can be restricted by non-competitive inhibitors of the same structure as the cofactor. The inhibitor will prevent the enzyme from changing the shape of the active site. 2. Enzyme cofactor or activator is part of the enzyme in the form of non-protein compounds. Cofactors can change the shape of the active site so that can be attached to certain substrates. Types of enzyme cofactors. A. Coenzymes are cofactors in the form of organic compounds or of vitamin, which are non-covalent bound to the enzyme. Examples: coenzyme and A. The protestic group is a cofactor in the form of inorganic compounds or minerals that are covalent bonded to enzymes. Example: Cl-amine and Ca2+ in amylase enzyme, Fe in hemoglobin, and Mg in chlorophyll. Enzymes that have bound to cofactors are called holoenzymes. The active site can be disrupted by competitive inhibitors having the same structure as the substrate. The inhibitor will prevent the substrate from binding. The allosteric site can be disrupted by non-competitive inhibitors having the same structures as the cofactor. The inhibitor will prevent the enzyme from changing the shape of the active site.
characteristic of one person of first of proteins. All proteins are equal and as it said that the enzyme only change the rate of reaction to a lower reaction specific enzyme as specific substrate from product as well. This is of synthesis case that have special set. So they only open one block, for example, unless enzyme that only work as substrate form of starch. Or enzyme can be used repeatedly or reused both, as long as the enzyme is not damaged. The enzyme is over and over again because it does not participate in the reaction. But enzymes do not turn into products, although the enzyme works to convert substrate to product. Enzymes don't turn into products instead. 6. Enzyme reaction is reversible. An enzyme can carry out two-way reaction, namely from substrate to product or product to substrate. Okay, how enzymes work? It is explained in two theories. The lock and key theory. Check the theory. First one is the lock and key theory. According to this theory, enzymes and substrate are considered as locks and key. According to this theory, an enzyme only works for one side of substrate by binding to the active site. The second one is implicit compatibility theory. According to this theory, the cofactor or activator of the enzyme will bind to the allosteric site. The cofactors change shape of the active site in order to bind the certain substrate. The substrate is then converted to products and released from the enzyme. And the last enzymes can be reused for subsequent substrates. Factors affecting the work of enzymes. A. Effect of concentration of substance associated. First, enzyme concentration that is greater than the substrate will accelerate the reaction rate or accelerate formation of product. 2. Substrate concentration that is greater than the enzyme will result in saturate substrate concentration or maximum reaction rate, which causes uncatalyzed substrate. 3. A large concentration of the will slow down the reaction or inhibit the formation of products. The way to prevent from inhibiting product formation raise the concentration of enzyme factors and substrate. Temperature affects the work of enzymes. First, the higher the temperature, the kinetic energy of the substrate and enzyme increases, making it easier for them to bind each other. 2. Enzyme activity increases at optimum temperature or maximum temperature about 40 degrees Celsius. 3. Temperatures that are too high or 40 degrees Celsius cause enzymes do not work as the structure of the enzyme is changed due to protein denaturation. Denatured enzyme cannot be used. 4. Temperatures that are below 30 degrees Celsius cause the enzyme to not work because the enzyme is inactive. Enzymes that are inactive can still be used if the temperature returns to normal. C. pH can affect the structure of the protein on the active site, so that the substrate binds. And the last one, the optimum pH of the enzyme is different. Enzyme is not at the optimum neutral pH, slightly alkaline. The enzyme peptidoglycan works at very acid pH, and most does work at an alkaline. The enzyme name before nomenclature. Enzyme were made by 1. Carelessly when found, example trypsin, pepsin, and guanine. 2. FAs to the substrate name, example glucose, react, phase, acid or fake, amylase, starch, yes, protein, and lactase, lactate. And the base on the type of reaction catalyzed, example, the hydrogenase catalyzed and then best reaction. Hydrolyses are enzymes that catalyze the hydrolysis. Example, this enzyme such as amylase, sucrase, trypsin, thymine, protein, etc. 4. Gas is an enzyme that breaks the chemical double bond on the substrate. Example, carbonic anhydrolase and ornithine decarboxylase. 5. Isomerase is an enzyme that catalyzes the formation of isomerism of a single compound. Example, protein sulfide isomerase.